Good afternoon, everyone. Welcome to our Lunch and Learn webinar. We're gonna wait just a couple of minutes to let folks join and then we'll get started. All right. Hello, everyone. Welcome to today's Lunch and Learn webinar on the Ovation Research Network. I'm Barry, co-founder and CEO of Ovation, and I'm here with my colleague, Pauline, our Vice President of Clinical Affairs. We're glad so many of you are able to join us today, and we're looking forward to sharing this exciting opportunity with you and to answering your questions about the Ovation Research Network. Before we get started, I'll tell you just a little bit about Ovation. We're a technology company. We started in the healthcare space in 2017. We're backed by leading life science and technology investors. My co-founder and I came from scientific research and life science backgrounds, but we saw an opportunity in the clinical space to shorten the distance between clinical data and research, specifically when it comes to making precision diagnostics more accessible. Today, we build software that helps independent molecular diagnostic labs run their business through our LIMS offering. But perhaps more importantly, we build what we're here to talk about today, the Ovation Research Network. I'm really excited to tell you about that research network now. Just a little bit of an agenda. Today, we're going to cover the unrealized promise of precision medicines, why clinical laboratories are key to the future of precision drug development, and how the Ovation Research Network enables labs like yours to take part in and benefit from a new sustainable revenue stream while doing good in the world. Just a couple of housekeeping items. A recording of today's webinar will be available on demand within 24 hours. And please ask questions. Today is for you. All of your questions will be answered either during our Q&A session and during the webinar or via follow-up. So please don't hesitate to ask questions as we go. You can use the Q&A tab at the bottom of your window to ask questions. And my colleague, Matt, will be curating those questions and making sure that we get to as many as we can during the webinar today. Okay, let's start with the promise of precision medicine and a challenge that healthcare has in bringing that promise to patients. Precision therapies are life-changing, selected based on patients' personal genotype and phenotype, Precision therapies offer improved outcomes, reduce complications, and in some cases, dramatically extend survival and quality of life for patients with complex illnesses. Molecular diagnostic labs clearly have a foundational role to play in the delivery of these precision therapies. But you may be noticing, as we have, that not all patients who could benefit from precision therapies are getting them. Many among us have acquaintances or family that could have benefited from precision therapies. This is an issue that touches all of us both personally and professionally. So why don't these life-changing therapies make it to the patients that could use them? Well, there's a chicken and egg problem. Patients don't get access to these therapies, or at least don't get those therapies paid for until they're proven economically effective. And payers can't determine the economic effectiveness of precision therapies until more patients receive them. And sometimes these therapies' benefits extend over many years. Hence, a chicken and egg problem. And these therapies are expensive. It costs more than $1 billion to develop a new therapy and the precision diagnostic tests that unlock its use. And the result is that without payer engagement, it's very difficult to generate broad access to these therapies, even for patients that would benefit from them. One of the causes of the cost of developing these therapies is the cost and time associated with clinical trials. One way to accelerate this process is by discovering biomarkers that can help select and enrich patient cohorts for clinical trials. So a prognostic biomarker indicates which patients are likely to develop a targetable disease. So discovering that biomarker, developing and validating assays, and deploying those assays into clinical trials allows life science researchers, biotech and pharma companies, to perform clinical trials for new therapies faster and more efficiently. The result may be accelerating what is a very long, sometimes up to 10-year process to develop a new medicine. But even these biomarker discovery and assay validation steps, even they often require their own clinical trials. And what we're just showing here is an example of one of these biomarker discovery trials. The steps here are to identify, recruit, and enroll patients, to collect specimens from those enrolled patients, and to perform characterization of those specimens, to identify biomarkers, genomic or otherwise, that predict which patients are likely to develop a disease or progress through that disease. The result of this clinical trial would then lead into accelerating additional clinical trials for therapeutic development. This is a long process, and it gets even harder when the therapy at the end of this is a precision therapy. As we're showing here, only a small fraction of enrolled patients will qualify. And it's harder still when the disease in question has a slow or rare progression. Even fewer patients will progress or develop the disease within the time of a clinical trial. This is the underlying reason why precision therapies are so expensive, and one of the reasons why they're so hard to get into the market where they can help patients. Winston, my co-founder, and I, and many others in the industry, from patients to labs to researchers, know that there is a better way. Mountains of data are collected every day by clinical activities. If only we can get this real-world data to the right people at the right time, we can reduce the time to market for precision therapies. The core of biomarker discovery and validation is analysis of clinical genomic data that captures the association between biomarkers and disease diagnosis and progression. Real-world data provides faster access to answers for doing these kind of association studies. Querying a database is much faster than recruiting patients, enrolling them in a clinical trial, and only then being able to make a biomarker measurement. If we could produce clinical genomic data sets that could be queried like a database, instead of measuring clinical trials, we could accelerate the development of precision therapies. We could get them into the hands of patients faster. Real world data seems to hold the promise of making that possible. But important data for those analyses, such as patient level genomic data, is missing from typical real world data records, delaying the discovery process. As many of you know from your experience in labs, structured genomic data rarely ends up back in the clinical record, in the electronic medical record that can be mined. But there's even a more subtle point. Discovery of novel biomarkers and validation of novel assays for those biomarkers, by definition, requires characterization that is not normally done in the clinical flow. In other words, the most valuable data is the data that isn't yet collected. To achieve the full potential of real-world data, to accelerate discovery and development of novel therapies, the next evolution, the next revolution in real-world data and precision medicine will have to bring together phenotypic data in the electronic medical record, structured laboratory molecular data, and novel genomic characterization. Only together can we advance the development of biomarkers, the development of new assays more quickly. And labs like yours hold the key to this next evolution of real-world data. We built the Ovation Research Network to bring those three components together. Existing real-world data, measuring patient phenotype, diagnosis, and disease progression, and working with diagnostic labs to add structured laboratory data and genomic data from research characterization of de-identified remainder samples, all linked together in the de-identified domain to accelerate research. And we're doing just that. We're able to accelerate biomarker discovery trials, assay validation, and patient recruitment. These steps by as much as tenfold for researchers in life science. Researchers like Carlos Bustamante. He's a professor of biomedical data science and genetics and biology at Stanford University. And he's the CEO of Galatea Bio. With data from the Ovation Research Network, Galatea Bio is able to accelerate what is traditionally a multi-year genomic biomarker project into just months. This is truly game-changing for companies like Galatea Bio. Imagine if we could help every biotech company accelerate their program by tenfold. Imagine the rate at which we could help new discoveries become new therapies. The world of biomarker discovery and development will never be the same. And your lab can hold the key to making this happen. But sharing data and participating in this evolution of real-world data is not without challenges. Labs face a number of challenges in sharing data for real-world research use. First, they need a legal consent framework for gaining patient consent for research testing, and a clear understanding of the relationship between the clinical flow and research use of de-identified samples. Labs need resources, business development and sales resources to develop relationships with life science researchers, to develop the business opportunities to make their data useful. Labs need sufficient data to answer research questions. Because many targeted therapies are relatively rare in the population, it's difficult for individual labs to have sufficiently large data sets on their own to meet researchers' study requirements for one, let alone many studies to produce business. Labs also need informatics infrastructure to collect, store, de-identify, and share this data securely and easily. And finally, none of this happens unless there are the specimens available for further characterization. So labs need the resources and effort to stand up biobanking operations and to do additional characterization of samples. All of these are challenges for clinical labs that are working every day under pressure to deliver clinical results to patients with fast turnaround times. There's a lot of work to do, and it's challenging for individual labs to get it all done. This is exactly why we built the Ovation Research Network and why we evolved our business to help diagnostic labs realize the opportunity from real-world data. Ovation brings remnant samples and data from independent diagnostic labs together, creating one of the world's most comprehensive and diverse clinicogenomic databases. By joining the Ovation Research Network, your lab gains infrastructure and scale needed to mobilize value in your remnant samples for an extensive variety of research projects and generating additional revenue for your lab. And we do all of this together, Ovation and the network of labs being successful. But here's how your lab benefits. First and foremost, our labs in the network today find that it's valuable to be at the forefront of new treatments and new medicines. Most labs know that their leftover samples carry value, of course. And it's frustrating not to be able to unlock that value, both from a business perspective and more importantly, from a scientific perspective. So participating in the research network gives you a chance to mobilize all of that value by working together with other labs. This mission also resonates with providers and their clients, allowing labs that participate in the Ovation Research Network to differentiate themselves in a competitive market. Your ability to say that you are safely and ethically using de-identified samples to help advance new therapies, to know that you are a socially responsible and forward-thinking lab is something that makes a difference to providers and patients when deciding where to send their clinical samples for testing. The second piece, of course, is that it allows your lab to add a new revenue stream. So at a high level, here's how it works. You'll send your leftover samples to us. We pay the costs, labor, and materials for you to box those up, ship them to us, and under a business associate agreement, we'll turn them into research-ready samples through a de-identification process. We put those de-identified samples into our biorepository and store them and cover the cost of that storage. When a life science customer comes to us with a research need, we identify data that's already in our database or research samples that are relevant to answering that question. And at that point, we pay your lab in two ways. If we use your samples to perform additional research needed by our life science customer, we pay you a per sample origination fee. The other way we can generate revenue for your lab is by paying you to do add-on research testing, sending the sample back to your lab for you to perform additional characterization if you have the capabilities and capacity to do so. This allows your lab to win research business that would not normally be available. And we do this at your rates, of course, generating a revenue stream that doesn't rely on the reimbursement of individual test billing. And we believe we can bring a significant amount of revenue to your lab over time. We're just getting started, the number of research projects, but as the research network grows, we believe this is a long-term lucrative partnership for your lab. Some participants in the Ovation Research Network are generating revenue that helps them justify acquisition and implementation of new instruments. They'll reduce the cost and increase the throughput in the rest of their diagnostic business. Participating in the research network helps increase the return on investment in acquiring those new instruments, developing those new processes. Finally, if your lab is performing internal R&D, there's an opportunity we can discuss, depending on sample quality and some other factors, about accessing aggregated and de-identified data from our clinicogenomic database at a much more favorable terms than it would be for outside researchers, helping your lab accelerate your own internal research and development. So this time, I'm going to turn it over to my colleague, Pauline, our VP of Clinical Affairs. She's going to tell you a little bit more about the details of how we make all of this happen.
So we understand that the people in the lab and labs in a clinical workflow are really busy and have the focus of patient results uppermost in their mind. So we've actually allowed for this when we are looking at your remnant specimens where you would normally just put them in a biohazard bag. We're asking that you send them to us instead. In this case, we have a whole team mobilized to help you ensure that the shipping of these remnant specimens to our biorepository is as effortless as possible and have the least amount of impact on your clinical operations. In addition to that, we want to make sure that you guys are compensated for that extra work. And then Ovation has actually spent a great deal of effort and time to handle the de-identification within that legal framework that Barry mentioned earlier. And so we will actually help you de-identify the specimens, or the biorepository will actually de-identify the specimens for future processing. This allows us to categorize and inventory all of the specimens that go into a biorepository so that when it comes to pulling them for research studies, we have a very organized inventory of these. So when the researchers ask for that data set, we can actually pull from the cohorts really easily. And then finally, what we would like to do is actually work with you to get research testing up and running in your lab where you have the capabilities. This will actually help you fulfill any unused capacity in your lab and hopefully create a pipeline for your future clinical testing. There are three, the next slide, I think, Barry. There are three things that we need from you in order to participate in the network. We really would like you to adopt Ovation's unified patient consent. We will work with you on that. We understand the clinical workflows and the consenting universe in both your clinical workflow and what we're asking for in our Ovation Research Network. We would like, obviously, if you're going to be sending us samples to our biorepository, to actually stand up a really easy and standardized biobanking operation where our team will actually help you get the samples organized and ready to ship, and that we have a beginning to end operation where we can receive the samples and inventory them for pulling them out as research cohorts. And then finally, we are more than happy to work with you to qualify your lab for research testing. We have several of these studies underway and very successfully getting very good research data. So if you are chosen to do research testing, we will come in and actually qualify and help you set up the research testing and validate that testing before we begin.
Thanks, Pauline. I'm going to cover a couple of common questions. I see that some folks have asked these already, so I've prepared. I'll cover a couple of common questions, and if we need any more time, we'll take some time for additional questions after. One very common question we get is what consent needs to be in place to work with the Ovation Research Network. So expanding a little bit on Pauline's description, our experts in the clinical affairs group will help your lab adopt a consenting strategy that works for you based on what you have in place already. In the United States, HIPAA does not require consent for use of de-identified samples and data in research. That means that the medical consent you're receiving to do clinical testing on samples today is acceptable as a means of consent, as long as the language does not explicitly preclude research testing. And most medical consents already go above and beyond and explicitly state that patients' de-identified data and specimens may be used for research purposes. We do believe that notifying patients about the usage of their remnant samples is the right thing to do. and is an important part of making sure that the network is beneficial to everyone, including the patients, that they understand that they're participating and what the benefit is going to be ultimately to the world. So we do that within our research network labs by getting the unified patient consent language adopted. We've reviewed this language with legal experts specializing in patient data privacy and with a number of independent IRBs. And our team will work with your lab over time to get this language into your standard test requisition forms at a pace that makes sense for your lab. When we've done this, we found that providers easily understand the details of the consent and are not just comfortable, but excited about the opportunity to add this notification to the language, to talk about it with their patients, and to engage them in the research process. You may have talked to biorepositories in the past. So how is the Ovation Research Network different from other biorepositories? The answer is that Ovation and the Ovation Research Network is different from other biorepositories in one major way. We're not selling specimens directly to the life science industry or healthcare. We're creating a clinical genomic database that allows us to provide data to answer a large variety of questions within the space. A health industry researcher will come to us and say, I'd like to measure the genomic profile of a number of patients in a specific disease cohort. We say, great, we've got that data, or we've got specimens in the freezer. We will pull them out. We'll send them back to the originating lab if possible, and we'll do whatever testing is needed for the project. We have the labs characterized as fully as they can, and that data becomes part of our data set that allows us to answer that life science researcher's question. That database query, the first time happens quickly because we already have the samples available. The second time it happens almost instantaneously because the data is already available in the data set. It's because of this strategy that working with us is so attractive to life science industry researchers and labs alike. For researchers, they come to us for a wide variety of projects across disease areas, and they know that they can get answers that are powerful because our data is coming from labs all over the country with a diverse population, not only demographically, but also therapeutically. Finally, for labs, unlike working with potentially other biorepositories, with the Ovation Research Network, they're able to first get paid for origination for samples that other biorepositories wouldn't typically be interested in, since our model allows us to collect specimens of all types. Second, labs actually get the opportunity to participate in this market by performing the additional research testing. And finally, our clinical affairs experts work with labs to ensure that their research teams and research testing is validated and that their biobanking operations are sound. So this is a chance for your staff to be educated on these additional areas and to build skill sets they would otherwise not have access to. In other words, this is not a transactional relationship. This is a long-term partnership. We've created the Ovation Research Network for the long term because we think it's the right way to accelerate life science research. Last, you may be wondering, what resources are required to participate in the research network? Ovation is a LIMS producer. Do you have to use our LIMS in order to participate? We say first, the overarching goal is that it is zero cost for your lab to participate in the research network. We want this to be a net positive, both business and scientific win. We understand that labs are short on resources, perhaps even more so right now. And we've all been through a long couple of years and months and years. So we want to make sure this is low impact to you. And we'll try and make it as close to take your remnant samples, throw them in a box and ship them to us. Try and make it as close to that as possible. We've standardized the collection and cataloging process through software that we'll set up for you. And that software actively ensures that shipping is seamless and straightforward for your lab. As I stated earlier, Ovation pays the labor and materials cost to get your lab's remnant samples from the end of your clinical workflow into our biorepository. So we'll handle the training of your team and best practices to ensure high sample quality throughout storage, proper documentation, and shipping steps. You do not need to use the Ovation LIMS to participate in the Ovation Research Network. Like I said before, we do have software that will help you get in place to serve as the informatics infrastructure for the collection, cataloging, and shipping of the specimens. But it's okay if your LIMS is separate from Ovation. Of course, things are a little bit less complicated if you're using Ovation as your LIMS software as well. So we encourage you to use it for your molecular testing, but we know for a variety of reasons that's not always possible. We would love you to participate in the Ovation Research Network anyways. At this point, I know we're coming up on our time limit, but Matt, I turn it over to your direction if there's additional questions or if we want to finish up here.
Sure. It does look like we can squeeze in maybe one question here. I believe Umar asked, what's the cost of sample storage and what types of samples, infectious diseases or human genomics?
Wonderful questions. So there's no cost to the lab to store samples. As I just stated, the Ovation Research Network covers the labor and materials cost to get those specimens transported to our biorepository, where we de-identify them and we cover the cost of storage. Zero cost to your lab. You'll only hear about when the samples get used in research. There's a revenue stream coming back to you. First question. Second question, what types of specimens are we interested in? The answer is virtually all specimens that contain genomic DNA and RNA are of interest to us. We have a number of labs that are doing infectious disease testing. We have a number of labs that are doing germline. We have a number of labs that are doing somatic genomic testing. All of those biospecimens are of interest and useful within the Ovation Research Network. Again, the model here is to cast a wide net to be able to capture specimens from the broad variety, the broad heterogeneity of patient populations and therapeutic areas.
I have one more question for you, Barry. What types of testing do you outsource to labs for the Ovation sponsored research?
Another excellent question. It depends a little bit on the research questions asked by our life science researchers. Our desire is to characterize these samples as fully as possible when we pull them out of the freezer. We typically do total nucleic acid extraction from those samples, so collecting both DNA and RNA libraries. Very often we're doing NGS, either whole exome or whole genome sequencing on those samples, and RNA-seq if possible. In some cases, we're also doing targeted characterization using qPCR or other methods. Really, any lab that's doing molecular testing today potentially has the capability to do at least some of the research testing that we're doing within the research network.
Have another one from Gabriel as well. How about LCMS for metabolimics? Hopefully I pronounced that correctly.
Yeah, another great question. In fact, we do have a number of researchers that are now starting to ask about metabolomic studies and biomarker characterization. Get in touch, we'd love to chat.
One more question from Stephen. Is there any benefit to Ovation to receive genomic data as opposed to the actual specimen?
Great question. If the lab is already characterizing the genomic data as part of the clinical flow, yes, we'd love to work with you to capture that data, even if the specimen isn't available. In general, we do have a number of both metadata and quality gates associated with the genomic data, and potentially also with the bioinformatics that we'd have to work through to make sure that that data is combinable with the rest of our clinical genomic asset. But if it's possible to capture the genomic data and the specimen, that only adds to the value of the specimen itself. I do know that one other question that comes to mind, Matt, if you don't mind me jumping in. I know we've got some folks from outside the US here on the webinar. We do accept specimens from outside the US. And we do this, we'll work with you on a case-by-case basis. We need to make sure that the consent framework and legal framework that we described here is applicable in your jurisdiction. But we do have collection outside the US, and it's of great interest to us.
Great. And just one last question, and I'll just reference that we'll follow up with you directly, Gabriel. Specific question around whether we integrate with Agilent. We'll follow up with you directly regarding that. We integrate with a whole host of instruments, so we'll follow up with you. And I think that's it for the Q&A, Barry.
Great. So, hope you're interested. Great to have you all here. Fantastic questions. If you're interested in participating in the Ovation Research Network, please get in touch with me directly or with anyone at Ovation. The next steps here, we'll review our network participation agreement that covers your participation in the network itself. We'll have you meet with our clinical affairs team to start understanding the biobanking process and SOPs. And together then, we'll define a work order, no cost to you, but it will lay out exactly what we're going to do to get you all set up and running, any actions we require on your lab side. Those two get signed together and then we begin the implementation. It typically takes 1 to 2 weeks for a lab to get up and running with the biobanking process and participate in the Ovation Research Network. With that, thank you all for joining. Really glad you were able to make it today. And you have our contact info here. We thank you for joining us, and we look forward to talking with you all again soon.